In this video, I'm just going to give the briefest of introductions to this idea of nutritional immunity. And so this is the way that our bodies sort of try to sequester nutrients away from pathogens that infect us. And so if we look at this, so we're looking at nutritional immunity. And like I said, this is going to be just the most cursory of introductions to this. So if this is something that you find interesting, there will be links to the references in the description down below, and so you can look into that further if you want. But in this one, I'm just going to sort of briefly introduce us to it. And so the first thing I want to discuss here is what is called a siderophore or siderophore. And so these are molecules that bacteria actually use to grab iron out of solution, or even away from like heme or things like that. And so what we see here is we have our host cell here, which wants to take up this iron from the lactoferrin, from this LCN2, which is the lipocalin-2, which I'll talk about here in just a second. And so the bacteria, which we have over here, send out these siderophores in these what they call stealth siderophores, which I'm not going to really get into here, but those actually take up the iron and actually bind to it extremely tightly, and then the bacteria can actually take those siderophores back into themselves. Well, this LCN2, this lipocalin-2, is actually a defense that our hosts, or our body cells, which we are the hosts, so those are our body cells, actually use to try and take the siderophores back into themselves. So the bacteria let out the siderophore, which binds to the iron, and then this lipocalin-2 actually binds to the bacterial siderophore, which is bound to iron, and then brings that back in. And so we can see that over here where we have this LCN2, this lipocalin-2, and it can undergo these conformational changes, and then it can actually take in one of the siderophores here, and then that can actually be taken back into the human host cell. So what I have over here on the side are just some of the examples of the different kind of siderophores that bacteria use. So we have these carboxylate, the catecholates, hydroxymates, and phenolates here. So this is a particular carboxylate type right here, which we see in Staphylococcus aureus. We have these catecolate types here, the phenolate types here, and then these mixed types here, which we see can have multiple different kinds of these functional groups on them. And so these are just some of the siderophores that bacteria use. And so one of the things that the human body can do to try and sequester the iron away from the bacteria is the release of this hepcidin, which I discussed in the video on iron. In fact, we looked at this figure right here where we see this hepcidin actually blocks this transporter here that is supposed to move iron out of the cell and into the bloodstream. And so the hepcidin, which is a 25 amino acid peptide hormone, is secreted by the liver in response to inflammatory stimuli. So there's a greater than 100-fold increase in urinary hepcidin concentrations in a patient who develops systemic infections. So this shows that the hepcidin exerts these effects by regulating iron homeostasis. So infection and the resulting inflammatory stimuli, particularly interleukin-6. So this is like an immune response molecule here. So that stimulates the hepcidin transport in the liver transcription in the liver through the JAK2 STAT3 signaling. So the hepcidin signals for internalization and degradation of the iron exporter ferroportin, which is what we have up here. That's the ferroportin. So hepcidin mediated declines in ferroportin inhibit iron transfer into circulation from the enterocytes, macrophages, so white blood cells, and iron storing hepatocytes, thereby withholding iron from invading pathogens. And from a different paper here. So, animal studies have assessed the liver transcript abundance of all 24 mammalian zinc transporters. So remember those zip transporters they talked about in the zinc video. And the zinc binding protein metallothionein in response to LPS, which is lipopolysaccharides, which is a bacterial endotoxin, or turpentine, which is an inflammatory stimulus injection. So they're injecting, lab rats, and then looking at what's going on with these zip proteins, so these ZERT-ERT-like proteins. So metallothionein and the zinc transporter ZIP14 demonstrated the greatest magnitude of change, increasing by 35 and 3.5 fold in LPS and turpentine injected mice respectively. And so those ZIP transporters, remember, are the ones that bring zinc in, where it was the ZNT that excreted the zinc back out of the cell. And so we get this increase in these ZIP proteins. So the cells are bringing zinc in even in much higher concentrations than in a healthy individual and somebody who doesn't have this inflammatory response. And so, yeah, that is again sequestering zinc away from pathogens. And so, yeah, that is, like I said, just the briefest of introductions to this idea of nutritional immunity. So, immunity by trying to starve pathogens essentially of these nutritions that they are trying to steal from the host. But anyway, I hope you found this video interesting. Like I said, if you do find this interesting and want to look into it more, then check out the description below. I'll link to a PDF that will have all of the references, and it'll have more references than what I even used here. So if you want to look into that, then feel free. But anyway, I'll see you in the next video.